Phillips, and today we're looking at genetic fingerprinting and the use of electrophoresis in genetic fingerprinting is. Well, this is obviously widely used in forensics, but it can be used in plant and animal breeding, medical diagnoses, and the whole purpose of this is it relies on the fact that DNAs of all individuals except for identical twins is unique, and therefore we can identify samples of relies on the fact that most human genome contains repetitive non-coding DNA sequences, and they have something called a variable number of tandem repeats. These have a unique length and pattern for everyone, but the closer related you are, the similar these are. Samples, we take the samples, we isolate the DNA, we may do lots of copies of them through PCR, and we do a process called electrophoresis, which gives us an image, okay, and then that image is put into a computer, which can then analyse the results. The DNA then, or what we use is something called gel electrophoresis. And gel electrophoresis uses something called restriction fragments, which have been separated using restriction endonucleases. And basically your DNA fragments treated with restriction endonucleases, you'll take a fragment and you'll split it into lots of different sections, different sizes, and this allows us to read DNA from an accuracy of 1 base difference. So it's widely used in gene technology to separate DNA segments, so we can identify them and analyse them. We use something called a gel plate containing a molecule called agar-rose, and the agar-rose is a very pure, like agar jelly that DNA can move through. It's also covered in a buffer solution, and the key fact of how this works is DNA has a slightly negative. We attach electrodes to either end of the electrophoresis tank, and we put a current through it. This places opposite charge on the electrode, either electrode, and we put the DNA, we treat it with restriction endonuclease to cut it into fragments, and then we put the DNA into the negative end of the gel, because DNA has a negative charge, so that will mean it will move away from the negative end and be attracted towards the positive end of and put it in buffer solution, and we leave the DNA in the solution for a set amount of time, which means it moves towards the positive electrode. Shorter strands of DNA get less resistance and less caught up in the agar-rose gel, so they'll move longer across than the longer lengths. And then positions can be identified using staining, which identifies where the DNA. So we use a process called southern blotting to get the DNA out of the gel, and basically something called nylon or nitrocellulose sheets are placed over the gel, covering in paper towels, and it's pressed overnight. The DNA fragments then become transferred to the sheet, and we can use radioactively labelled DNA beforehand to put it in a photographic film, and that will expose it, or we can use. So you can see here we do put it on there, we leave it in the dark, and then we DNA up. So let's just see how you split this up in an exam question. So how we can use it to then make genetic fingerprints. So stage 1, you would extract it, so you'd remove it from the cell and you would replicate it using PCR. Don't forget to talk about how you would extract it. You could use cell homogenification and ultracentrifugation from AS. Stage 2, then we digest it into smaller fragments using the same restriction endonuclease. That'll be for all samples. Okay, and then 3, we will then use electrophoresis to separate the DNA into different sizes using electrophoresis before transferring it to a nylon membrane. So with the nylon membrane, we would use hybridization, where we use DNA probes, and we can use that to identify where the DNA is, and then we can develop it on an X-ray film and identify patterns which should be unique to different